Um, Good afternoon, ladies and gentlemen. I would like to first of all thank the organizers of this webinar to update our colleagues in the third realm, as they call it, which are very essential stakeholders in our society to keep our society awakened and also be abreast with what they should know as far as life is concerned. I'm going to quickly, just like Dr. Egese had done, put you through jumping from a root tuber to a leguminous crop, cowpea, which we call popularly in Nigeria as beans. I want to show us the stages that we arrived at developing, the stages we followed and arrived at the development of a new insect-resistant cowpea variety called SAMP 20. SAM stands for Samaru, P for cowpea, which literally means Samaru beans variety number 20T, which calls transgenic. It's a collective work of many of us. The names are listed as you can see them. And I'm going to talk about briefly the historical perspective of this work, what led to where we are, and what are the processes we followed, including that which involved seeking for clearance from the National Biosafety Management Agency. As you heard, Dr. Rufus Egbegba has alluded to our work. Now, beans, as we call it, cowpeas, this is an American word, cowpea, we call it beans here. We all know how important it is. It does a lot of things for us, including the provision of very cheap protein, plant protein for us, among other things. The perspective why cowpea should receive attention is that Nigeria produces around 3 million to 3.6 million tons. We have also a good land for the production of cowpea, but topmost, Nigeria, cowpea or beans can grow almost everywhere in this country once it is managed very well. But it does well more in the savanna drylands. So the other good story about beans is that Nigeria is the largest grower or producer of beans. But unfortunately, Nigeria does not grow enough beans for its citizens. So we end up importing close to half a million tons every year to satisfy the need of our cowpea consumers. Now, why? Because average yield on our farmers' field is low, around 350 kilograms per hectare, with a maximum of maybe 500 kilograms of production on farms, instead of reaching around 2 tons per hectare. So you see there is a very wide yield gap. And what is the reason? The reason is primarily due to insect pest infestation, both in the field and also in storage. Among the insects, there is this culprit called maruca. This is a butterfly-like insect, but then in one of its stages of development, there is a caterpillar, which you will see on the left, on my left-hand side of the screen. This caterpillar feeds on cowpea reproductive organs, the flower bud, the developed flower, and then it burrows on into the pod, developing pod also. The result is that either you do not have flowers at all, which means your yield will be affected. Reduction has been recorded up to 90% yield reduction in farmers' field. So this is a very serious insect that affects cowpea production. Now, this problem has been on for ever since cowpea has been grown by farmers. Until the year 2001, international scientists gathered in Dakar, Senegal, including myself at that time, and priority research area for cowpea production was identified, and this insect called Maruca vitrata was pinned as one of the important insects that should be controlled. Now, it reduces up to 90% yield. It causes up to 90% yield loss, and then it can induce reduction of grain quality, and so reduces farmers' income at the end of the day. Now, how has it been controlled? There is only one way: to be continuously bombarding it with chemical insecticides, spray them as we used to do. Now, this sprays often sometimes to get a good cowpea crop, you might spray up to eight times in a season before you will be able to get some reasonable yield as a result of this infestation by this insect. Now, there are two bad things about these chemical insecticides, or two or three. One, they are very poisonous and they can easily cause fatality in humans or even affect their health. They are expensive, and also when they are released into our environment, they might go and pollute our water bodies. So it is not a sustainable method of controlling this insect. However, the most inexpensive and safe method is to develop host plant resistance. That is through development of self-resisting variety which can protect itself against the insect. Now, this approach was followed. For you to develop a variety, you need a parent that will give you the genetic material from which you can develop this new variety. But we have checked up to 15,000 different types of cowpeas, including relatives of cowpea, in order to identify source of resistance. None was found to be resistant. So some other approaches called wide crossing. Crossing is a technique used in combining, in mating male and female, so that you get an offspring that will give you a character that will be inherited from both parents. You know, it's kind of a selective marriage between a groom, a selected good performing groom, and a good performing bride in order to get something that is good. So that's what we do in cowpeas. But you can only get a potential groom that has those characteristics, and then a potential bride that has the characteristics. But the 15,000 grooms and brides of cowpeas, if you may call it, none of them have this character. And yet we have this very important problem that we must tackle. So in recent times, science got developed to a level that you can deploy genetic engineering and a different approach from conventional ways of developing varieties. Here is how it goes. You take a cowpea and then get a source of a gene, what is called gene. A gene is a factor, a heritable factor, which we put in either plants or animals, and then this factor will enable the plant or the animal to be able to do certain things, like protecting itself from a disease, or like producing more sugar so that it becomes sweet, or it can produce more oil if it is concerned with oil production and so on. So science has made it in such a manner, as you have heard from Dr. Egese, that you can get genes now and then put them from one source to another source, which means you will now be, as opposed to the earlier explanation where we develop new varieties conventionally, that is by marrying brides, which are brides, mean you are talking about both of them human beings. But in this new approach, which is called genetic engineering or biotechnology, you can now marry a human being with a cow and then get an offspring out of, kind of. You know, so this provides a novel opportunity the world over. And this approach has been utilized for more than 20 years now. And new varieties have been developed all over the world. And one of the concerns about this approach, as you listen to Dr. Beba, is that people got concerned that are these varieties going to be safe for food? Are they going to be safe for our environment? For food means, are they not going to cause any disease? Are they not going to cause any allergy? Are they not going to cause any other harm to us? And then environmentally, will they not grow a simple cowpea? Will it not grow and overtake every other plant in our environment or become a tree like we have in some cases in some weeds that will be imported from some countries and then they will come and become a problem. So these are the concerns. Now, to be able to engage in this work, we had to seek for permission from NBMA that here is what we intend to do. We want to develop an insect-resistant cowpea variety through genetic engineering. I'm going to show you the procedure we followed. They came and looked at our facilities and checked everything and gave us a go-ahead and continued to monitor us when we keep on developing. So this is a cowpea. Then we put it in a container to put the genes inside it, and then later on in a medium in a laboratory and try to grow shoots out of the embryo. These shoots are managed and nurtured to this stage, and then it becomes a plantlet. Then it is conditioned, because at that stage, it is like under a too much comfortable environment. So you have to gradually expose it to natural environment outside the laboratory, which is called hardening. So after hardening, we take it to the screen house where it will come in contact with the environmental condition and then develop seeds out of it. You see one of these, they have developed seeds, and these are the seeds. We then said, these seeds have been tested by applying those larvae I showed you and selected and confirmed their resistance. Now what is left is we have to take them to the natural field under which they will grow and still check whether they will continue to exhibit this resistance to the insect. This is called a confined field trial. And we had to write an application to NBMA to give us the permission to conduct this. They gave us conditions to comply with. And from time to time, they come to follow up on how we conduct this research. And then we selected, these are our colleagues collecting data. And then we came up with one that is showing the highest performance. You can see 709A, which is the genetically engineered one, a biotech one. The one in the middle, IT86D1010, is unimproved. You see, it doesn't have, as a result of the insect infestation, it could not sustain any pods completely because the insects devastated it, it couldn't produce anything. From here, we continued this test of efficacy, and it is designed in such a manner that on each single plant, 40 larvae of that insect was placed. So actually, if any plant produces a flower and the flower survives, it must be resistant. Otherwise, it would have been eaten up. And eventually, the one of the line 709 was selected, which is on the left-hand side, 709, and then IT86D from the old plant from which the improvement was made. So this 709, it is as good as its parent material, IT86D1010. It is farmers don't grow this type. It is smooth seeded. The leaf is not good. Farmers are not used to it. So we have to now, but in terms of productivity, 709 is about 5 times more productive than unimproved cowpea. So because our farmers will not accept it, we have to initiate a breeding program in which we will now use flower-to-flower crossing and then put in these characteristics of resistance to Maruca. This is how the hybridization is done. In this stage, we use a flower of 709 and then brush it on a flower of another good material, but not resistant to Maruca, which is IT87 499-35, which is some P10, one of our good striga resistant material. So we transferred from 709A to some P10, IT97 499-35, the resistance to Maruca. And how have we been tracking the resistance? When you make a cross, you get several plants. Some of them may inherit the gene of interest, some may not. This is where we do it. We use a strip, a test strip. The presence of the gene will show 2 bands, two pinkish bands, which you'll see on the figure here. The one that is titled positive BT result, it has 2 bands of pink. And then the one that is not resistant will have only a single band. Any plant whose leaf or part showed double band, we know it is resistant. We don't need to even bring the insect to it. We know it has the gene. But later in the field, we will put the insect on it to confirm it again. So on that basis, we were able to identify 10 different families with this potential as shown in terms of yield per plant. Eventually, after the selection and at different stages of back crossing means marriage between parent and child, and then child and parent, and child and grandchild, and parent and grand-grandchild, parent and grand-grandchild, until that great-grandchild will become in all respects like the parent, except the new trait, which the parent doesn't have. Which means we were able to reach a stage where the new variety we are developing is as good as some P10, which is our commercial variety, but it is more than some P10 because it has the gene for resistance to Maruca. So we went on to different places outside. We started with Bakura, Minjibir, and Samaru here to test the performance of these new materials. These are the performance. This new material is called IT97KT. You will see how much it is producing compared to the parental material. For example, in Bakura, it has 210. The unimproved parent has only 210 kilograms per hectare, whereas the improved one has 388 kilograms per hectare. This is how the performance has been throughout. And with this, including some tests we carried out in terms of, is it any different in terms of its biochemical composition, its cooking ability, how palatable it is in local dishes and so on. The biochemical composition is an indicator of whether it is different from our normal cowpea or not. And all of this, in terms of crude protein, crude fat, proximate ash, carbohydrate, calories that it is expected, it is within the range of the control SAMP10. So this material IT97 KT, it is as good as SAMP10 in all respect, except that it is resistant to Maruca. We presented all this data in terms of food safety, environmental safety. Again, we conducted environmental safety where we planted the material. For every year, it will die just like any cowpea. Once it finishes flowering, it will die. So the places we have been planting it, it has not colonized anywhere more than any ordinary cowpea. So environmentally, there's no problem. Again, effect on non-target organisms. Is it bound to affect other insects that are beneficial? Our result shows that it is not any different from any ordinary cowpea, except that it is resistant. So we tendered this data to NBMA. Before now, we conduct all our evaluations under confinement because we are not allowed to take it outside the confinement. And the confinement means we can't take the material out for either utilization or any other thing or sell it to farmers until NBMA saw the evidence together with their monitoring visits and decided that this new cowpea, even though it emanated from biotechnology process, it is not any different from our ordinary SAMP10 cowpea. So next stage is we have to release it as a variety. The National Variety Release Committee has to be convinced with its agronomic potential also. Agronomic potential as well as the insect resistance potential. So we planted in several places. You see those locations where we planted them, including Ile-Ife, Makodi in Benue State, and so on and so forth. So the result indicated again, IT97 KT PBR on the average has the highest productivity compared to including even our most recently released commercial variety SAMP16. Again, this is an additional data. Now, this is a stability analysis. Stability analysis means, is it going to perform across locations or is only going to be effective in some locations and not others? Our analysis showed that it can be grown anywhere in this country. And so we have to also show that under farmers management, the previous slide I show you were for on-station scientist management. But so we handed it over to farmers and then they grew in the following locations, 20 locations and 30 communities and 27 local governments in Bauchi, Kaduna, Kano, Zamfara, and so on and so forth. So this here, the plot size is now much bigger, unlike the previous one where we have 4 meter, 4 rows. Here, the farmers managed trial is 10 rows by 10 meters. Okay. The condition under which farmers and also the scientists grow this cowpea is, unlike conventional cowpeas that requires up to around 8 times spray, this new cowpea variety, which was being developed, will give you more yield than the conventional one, which was protected by 8 sprays with only 2 sprays of insecticide. And the 2 sprays are targeted so that they will control non-Maruca insects. No, there are thrips, there are sometimes even aphids and sucking bugs. These are non-Maruca. Now, one may ask, then if there are other insects, why didn't you control all the other insects at once? Yes, science has its own restriction. You use what is most impactful and face that problem. Maruca occupies a very critical position in the life cycle of beings. It is the place where it feeds on flower buds and all reproductive structures. So if it eats away this, any other insect that might come, no matter how corrosive it is useless. It will not find anything. So that is why we targeted on Maruca and felt strongly that if we remove that 90% yield loss, the other 30 or 40% yield loss can be protected using these two sprays, which I alluded to. So here are the results of the farmer-managed trials called on-farm. For example, in Bakura, it produced 1.578 tons of grain and 800 kilograms per hectare of grains in Minjibur and 2.007 tons per hectare of grains and so on and so forth. So all over the places where it has been grown, this new variety outperformed our commercial variety. And we showed these evidences to the Variety Release Committee, including an assessment of some other qualities of the variety by men and women farmers. And the overall, this new variety compared to our commercial varieties scored the highest ranking, which means farmers are ready to accept it in terms of high grain yield, high pod load, early maturity, Maruca resistance, which is the principal one, less insecticide sprays, no visibility of damage of Maruca. So those are the criteria used by the women. So these are some of the farmers interacting with my colleague, Dr. Lawal Umar. You see one of the benefits is the lack of the striga, while on the conventional ones, you see striga. These are the descriptors for this new variety. It is now called SANP20. These are its other attributes. This is a typical SANP20T. You can see it is not different from any cowpea or beans plant. Those of you who have ever visited beans farm. So this is where it is on the left-hand side and on the right-hand side at different stages, early flowering and full podding. And then on the extreme right, it is when it is at maturity stage. This is one of the on-farm trials, evaluations visited by the contingent of the National Variety Release Committee, and they saw that this new variety has a very high potential, even under farmers. You can see the damage that was recorded in one of the commercial varieties, and then there was no, the damaged one are from the conventional variety, the undamaged one in either case is from the new variety. As I said earlier on, in terms of its local utility value, this new variety can give you as good moi moi, or if not better, as our SANP10 and also Akara. These are some honorable ladies who are helping themselves to the new products developed in the palatability test. On the basis of earliness, relatively high yield, which is between 2.5 tons to 2.9 tons yield potential, even under intense Maruca infestation, due to resistance to the insect, the variety SANP20 was registered and released to farmers in Nigeria on the 12th of December 2019. Now, we are at the stage of multiplying the breeder seed and the foundation seed, and to be sold to different companies that will go. I may have to allude to the benefits. On other fora, we have done the economic analysis, and the value that we will get simply from conservation of funds in terms of insecticide sprays is close to 46 billion naira. And if you add the advantage in terms of yield, it is also close to about 16 billion naira annually if Nigerian farmers can grow up to a total of 1 million hectares. With a more sophisticated economic analysis, these benefits were found to be even higher than our own estimation. Thank you very much for the opportunity, and I'm very eager if our colleagues in the media will find this presentation useful so that they can ask questions. Thank you so much.